During the adhesion phase of platelet plug formation, platelets contact and stick to parts of the damaged blood vessel, such as collagen fibers of the connective tissue that lies under the damaged endothelial tissue. As the adhesion process continues, a single layer of platelets forms, covering the damaged portion of the endothelium. In response to adhesion, platelets change their shape dramatically, swelling and forming spiked projections that allow them to contact one another. Once activated, the platelets release chemical messengers, including ADP, serotonin, and thromboxane A2. In response to these chemical messengers, more platelets begin to attach to the first layer of platelets. As more platelets aggregate, they release more chemicals, such as ADP, serotonin, and thromboxane, aggregating more platelets. The cycle of activation and aggregation continues, and an increasing number of platelets gather at the damaged site. Within 1 minute after injury, this accumulation and attachment of large numbers of platelets results in the formation of a platelet plug.
Platelet plug formation is triggered by damage to the subendothelium of a blood vessel. During the adhesion phase, platelets attach to the damaged subendothelium. This interaction is partially mediated by GP2B3A, a glycoprotein on the platelet surface that binds to a von Willebrand factor, which is present on the exposed endothelium. After a single layer of platelets develops over the exposed von Willebrand factor and collagen, the platelets become activated, change their shape dramatically, and send out spiked projections that allow them to interact with one another. Activated platelets release a number of substances from their granules, including ADP, thromboxane A2, and thrombin. These chemical messengers recruit and activate additional platelets from the bloodstream through binding to their target receptors. Let's take a closer look at each of these receptors now, beginning with the ADP receptor P2Y12. P2Y12 is an important receptor that sustains the series of events during thrombus formation. P2Y12 is the major receptor to amplify and sustain ADP-mediated platelet activation. It also triggers secretion of chemical messengers such as thromboxane A2, ADP, and thrombin, and activates GP2B, GP3A. Thromboxane A2 is synthesized by activated platelets through the activity of an enzyme called COX-1. Binding of ADP to P2Y12 triggers the synthesis of thromboxane A2 by COX-1, and the result is changes in the shape of the platelets. Among the effects of thromboxane A2 is its ability to stimulate vasoconstriction in the arteries. By binding to the TP-alpha and TP-beta receptors, thromboxane A2 also causes platelets to change shape from smooth and round to having spiky projections through which activated platelets interact with one another. The thrombin that is released by activated platelets in response to the binding of ADP to P2Y12 binds to a variety of receptors, including PAR1, on the surface of platelets. PAR1 mediates the thrombin-induced platelet response and can trigger most of the thrombin-induced signaling and the subsequent platelet secretion and aggregation. Activated platelets do not bind to each other directly, but instead they bind to fibrin through their GP2B, GP3B receptors during the aggregation phase to form a loose plug. During the clot formation phase of hemostasis, a mass of additional fibrin threads form, trapping red blood cells and covering the site of injury.
An atherosclerotic plaque develops early in life and grows slowly over decades. Most plaques are asymptomatic, while others manifest stable symptoms over a lifetime. In the context of ACS, a thrombus is formed after the fibrous cap of a high-risk plaque is disrupted through rupture or erosion. In response to vascular injury, smooth muscle cells vasoconstrict or spasm to reduce blood loss for 20 to 30 minutes, allowing time for the next steps in clot formation to occur. Once activated, platelets swell, form spiked processes, and release chemical messengers to aggregate and stick to other platelets, forming a temporary plug. As more platelets aggregate, they release more chemicals, such as ADP, serotonin, and thromboxane A2, aggregating more platelets. Within 1 minute, a platelet plug is formed. After the platelet plug is formed, a series of chemical reactions causes the formation of fibrin strands to reinforce the plug during the process of clotting. Within 30 to 60 minutes, platelet-derived growth factor stimulates smooth muscle cells to rebuild the vessel wall. Fibroblasts patch the injured area, and endothelial cells regenerate the endothelial lining. Once healed, the natural clot buster plasmin dissolves the clot slowly over several days.